Good evening, everyone. Thank you for joining today's evening webinar on cleanroom compliance and practical engineering solutions. Today, pharmaceutical companies are heavily investing in cleanroom infrastructures, HVAC system, validation activities, and monitoring systems. But despite these investments, many facilities is still facing the problem of designing zoning strategies, pressure cascade planning, operational discipline, and airflow management. And if these issues not solved, they can directly impact on contamination control risk, product quality degradations, patient safety threat, and regulatory approval delays. So as per the Clean Room International Regulations, the Clean Room is focused on ISO 14644, EU GMP standards, and US FDA standards. Now, as per the Clean Room compliance and all things, not all practical pharmaceutical designs, ISO or practical pharmaceutical HVAC design, ISO defines cleanliness level. EU GMP defines operational clean room requirement, and FDA ensure regulatory pharmaceutical compliance. There are the three causes or three root causes why this clean room fails in every clean room infrastructure. The root cause number one is design and zoning error. One of the biggest reason for the clean room compliance failure start at design stage itself. As we seen, area should become cleaner step by step towards critical production zones. As per GMP and ISO 14644, contamination control must begin layout planning, not after construction or during validation. The common mistake in improper segregation of material airlock, personal airlock, and waste movement. When these movements cross each other, cross contamination risk increases significantly. In many projects, especially retrofit and expansion projects, movement paths are planned based on available spaces instead of contamination control logic. This latter creates operational difficulties and GMP compliance issues. The other major issue is improper planning of personal airlock and material airlock. These are contamination control barriers, but we use this for the regular entries and outwards. If these things are not properly controlled, the proper air direction will be displayed. Pressure differentials will be not controlled, and area entry it will be eliminated. Without proper airlock, contamination air can enter clean zones. Pressure cascade become unstable and particle migration increase during door opening. Where it goes wrong, if we see the overall problems, if we see the overall the strategy, this cleanliness not follows, contamination control must begin at layout planning, not validation. Personal, material, and waste routes cross increases cross contamination risk. Malls and pals, which we use as personal airlock and material airlocks, treated as entry rooms rather than contamination control barriers. The last but not the least, zoning plant around available space rather than GMP laws. As per ISO 14644, zoning, airflow direction, and contamination control strategy should be finalized during the design and construction stage itself. To achieve effective compliance, we will see the practical engineering solution for the design and zoning strategies. To achieve this all problem solution to achieve effective clean room compliances, the following engineering practices are essential. The point number one is design layout based on progressive cleanliness steps by steps towards critical zone. Point number two is install proper interlock doors in all airlocks to avoid simultaneous door opening and pressure losses. Point number three is the proper perform risk-based zoning during concept design stage. Point number four is use pressure differential monitoring system between critical zones. Point number five is avoid dead corners and difficult to clean areas in layout planning. Point number six is to maintain distinct paths of personal and material airlocks, waste and prevent cross contamination. Point number seven is verify airflow directions from cleaner to less clean areas via smoke pattern testing. Point number eight is to conduct smoke pattern testing during commissioning to verify airflow directions. Point number nine is plan sufficient space for gowning, material staging, and cleaning activities. And the last point is to coordinate HVAC process and architecture team together during initial design stage. A clean room does not become compliant only during validations, dear. All clean rooms first become compliant through proper engineering, design, planning, and then through correct execution, commissioning, qualification, and operational control. The root cause number two is pressure cascade failure. As we all know, the HVAC system is the heart of any pharmaceutical clean room controlling air cleanness, airflow directions, temperature, humidity, and pressure differentials. Treating it as a comfort HVAC is one of the most critical mistakes in pharmaceutical facilities management. One of the most common failures is improper pressure cascade design and poor monitoring strategy. And I'm showing you in the on the screen, and I should always move from clean areas to less clean area to prevent contamination entry. The core principle of the pressure cascade to maintain minimum 10 to 20 pascal of pressure based on the room classification. And how it works, higher pressure is cleaner areas, pushes air towards lower pressure areas. And why it matters, prevent contamination and air entry, reduces cross-contamination risk, ensure product quality, and help maintain GMP compliances. When pressure becomes unstable, contamination air can enter clean areas, cross-contamination risk increases, product quality can be compromised, and GMP compliances get affected. The common operational problems we see is poor air balancing, HEPA filter choking, frequent door opening, improper return airflow, and lack of pressure monitoring system. After showing all of these problems, we will discuss about the practical engineering solutions we can comply on the clean room for the pressure cascade. To maintain stable clean room compliances performance, certain critical engineering and operational controls be must always be followed through these key points. The point number one is pressure cascade. Maintain proper differential pressure. As I already tell all of you to pressure minimum maintain 10 to 20 pressure pascals between adjacent room. We will have to monitor continuous differential pressure in real time, detect deviation instantly, perform periodic air balancing and revalidations after modifications, conduct regular integrity testing to ensure filter efficiency, install business management system alarm for pressure deviation alert, and last, follow strict PM schedule for AHU filters and return air system. The clean room should remain stable during actual operation, not only during validations. In pharmaceutical facilities, HVAC is not just for cooling, it is a critical contamination control engineering system. The root cause number three is the poor operational and clean build practices. Thus, many clean room fails even before production starts due to poor clean build and operational practicals. If proper clean build protocols are not followed during installation and commissioning, the can accumulate the dust inside. HEPA filters may get contaminated or damaged. Particle counts become unstable during qualification. Another common mistake is to validating clean room only under at rest conditions, while ignoring operational condition is a major compliance risk. As per clean room qualification requirement, proper validation should include particle count test, HEPA integrity testing, smoke visualization studies, airflow loss velocity and uniformity testing, differential pressure testing, recovery testing, and temperature and RH verification. This all came under ISO 14644.3. The compliance fails only, sorry, the compliance fails only during regulatory audits, media field simulation, process validation activities. At that stage, correction become more costly and time consuming. Even well-designed HVAC system cannot maintain compliance without operational discipline. And to solve this all, the practical engineering solution is to follow the strict protocols of this some points. Clean bin protocols, strict protocols during installation and commissioning to prevent early contamination. And second point is to use the verified approval cleaning materials and solutions to clean the areas like 70% isopropyl solutions, DI water, purified water, and hydrogen peroxide solutions. And the point number three is to seal dust during the storage and installation to prevent dust entry. Point number four is to validate under operational condition, not just at rest. Point number five is we have to maintain the strict gowning discipline before entry into clean rooms. Periodic monitoring, regular preventive maintenance, and regular and requalification cycles. To conclude my all these presentation points, real compliance is a system, not a checklist, as we all see. Most clean room failures track back to ignoring zoning, electricity cascade, and professional discipline. True compliance emerges only when all few police function as one integrated contamination control system. If you follow a proper system to standardize the clean room, we have to follow a proper design, HVAC engineering, construction, validation, and then operation. If you follow this all standard as per the process and standard process, the contamination control will be product quality plus patient safety. Thank you so much for all your valuable time.